So the next stain that we will be doing is the endospore stain. Just a little background on this. So endospores are like structures that are made by specific types of cells, bacterial cells to be more specific, which allow these endospores to live and grow in harsh environments. The endospore staining must be done after gram staining. Because by itself, the gram stain isn't able to show endospores under the microscope. The endospores will look clear under the microscope in a gram stain. That's why endospore staining is used after the gram stain. This method of staining uses two kinds of stains, malachite green and safranin, to help differentiate endospores from the rest of the cells in the culture. This method of staining, the endospore staining, also shows us if our unknown bacteria has the ability to actually produce spores. Now, before you would begin this experiment, you would obviously go up and grab the materials that you would need, and for the endospore staining, you would need your heat fix slide, which is right there. Just to clear up, this would have been the other slide that was not used for the acid-fast staining. This is just the other slide that we used for the gram staining and, when we prepared the smear earlier on. You will also need a slide clip for this experiment. You will also need malachite green stain. This will be your primary stain. It actually will stain the endospores, if any, in the unknown bacteria green. And then we have our safranin stain. This is your counter stain. It will, it's supposed to stain the vegetative cells that are in the unknown bacteria a reddish or pinkish color. My steam bath isn't up here right now, but you would need one of those as well. Water bottle for rinsing. Bibulous paper right here. You would also need a stain collection vessel, which I do not have, but the purpose of it is to dispose of any liquids that we accumulate in the stain tray, which is this lovely thing right here. We would also need a biohazardous waste container, and as always, make sure you have a new pair of clean gloves on, goggles, and a lab coat. So the process of doing the endospore staining kind of starts out like the acid-fast staining. So what you're first going to do is you're going to grab your slide out of your slide box, like so. And you're going to take, actually, I'm going to first grab it and with the slide clip. There's a reason for it. And then you're going to rip a piece of paper out of your book of bibulous paper. This is your bibulous paper right here. And you're going to put it on top of your slide. Cover it like so, and once you've done that, you want to place the heat fix slide on the slide rack of the steam bath. Just like that. I'm going to put this down. So once the slide is over the steam bath, you can apply your first stain, which is your primary stain, malachite green, which is this right here. I'll apply it on to the slide. Want to make sure that you get the bibulous paper nice and moist. I'm just going to speed up time. Put that down, and what you're going to now do is just let your slide steam over the steam bath for about 10 minutes. For the endospore stain, the heat that is applied from the steam bath is actually considered your mordant for this experiment. The heat will actually drive the malachite green stain into the endospores. Make sure that while the slide is on the steam bath, you keep the blotting paper moist. After letting the slide steam for about 10 minutes, you're going to remove it from the steam bath using your slide clip like so. and then you're going to carefully remove the bibulous paper off the slide. And since the bibulous paper is now contaminated, you're going to put that in the biohazardous waste container. And now we're going to let the slide cool off. I'm going to put it on the slide rack that is on the stain tray, and then we will continue. After the slide has cooled off, pick it up using your slide clip, tilt it downwards, and what we're going to do next is rinse it off with water gently. The water actually acts as your decolorizer for this experiment. It will rinse off the excess malachite green stain. And then after you rinse it off, just put it back on the stain tray. And now what you're going to do is you're going to apply your second stain, which is your saffron stain. You're going to apply it to the smear for about 30 to 45 seconds. Now remember, the saffron stain is your counter stain. It will stain the vegetative cells that are possibly in the smear. I'm just going to speed up time. Once you've added the saffron stain, pick the slide back up using your slide clip, tilt it downwards, and we're going to rinse it off again with water gently to get the excess saffron stain off. Alright, and then once you've rinsed off all the excess stain, you're going to take your book of bibulous paper and you're going to put your slide in, and you're just going to gently rub and dry the slide off. And once your slide has been completely dried, you can view it under the microscope. Once you've viewed your slide under the microscope, put it back in the slide box for safekeeping. Then what you're going to do is pour any liquids that we accumulated in the stain tray into the stain collection vessels for disposal. And then once you've done that, you're going to move all your materials out of the way and clean your workspace using germicide to disinfect it. Then you're going to use paper towels to wipe it down and dry it off. Then throw those paper towels away in the biohazardous waste container. Then remove your gloves. Put those in the biohazardous waste container as well. Go wash your hands. And then put on a pair of new gloves, because now you're going to move on to the next experiment, which involves macroscopically inspecting and identifying your unknown bacteria.